What do the animal model of startle reflex and fear conditioning tell us about the human adaptative anticipatory response? This bad word is just for say we didn't use the surprise in animal models. We can't say the animal is surprised. So, firstly, I will present you just an adaptative processing and just one famous sentence: the human brain, it has been written, is an anticipation machine, and making future is the most important thing it does. For my presentation, I think I will start to introduce the emotion. After we talk about animals model and behavior models. I will present you just some experiments of humans versus animals, just because we can. So you will see. Sorry. And after I will present you just the new techniques and new possibilities in the animal models for understand different processing of fear conditioning and startle reflex. And I finished by a surprise. So the component of the emotion is three components: situational component, event of our situation at the origin of the emotion, and cognitive component, truth, belief, weighting. It's the interpretation in the event. After we can have the physiological component. It's activation of the limbic system, the autonomous nervous system, the frontal lobe. It's different structure of brain. So perspective on emotion in biology and more. It's I just want to present you just to show you the spectrum of the emotion on your line temporal. Why line temporal? Just because firstly we can distinguish different zones with representative theorists and researchers draw from the field of psychology. Basic is emotion. It's really the basis of emotion. After basic emotion, you have appraisal, psychological construction, and social construction. Sorry. And it's why linear temporal? Just because this article, it's Gross, Dr. Gross. I really want to say you have two components. You have generation, emotion generation, and emotion regulation, two times. We can see the first, it is not really break time, it's really linear and constructive aspect. In same things, we can see just the pictures can imagine the situation of emotion. You can see the basic emotion on the first left, and after the appraisal, it's more. You can see in the corner sorry, the right corner, the different activation of brain, and you can see the basic emotions. It's really short connection, not really complicated. But after in appraisal, it's more complicated, and construction, it's really complicated. And after for the regulation, it's just take the social construction and appreciate to the response of emotion. And I want to really take another style of presented the temporal line of emotion. We have the situation, and after the attention, appraisal, and response. It's really it's a really shortcut, but we need to shortcut for understand and another. In cognitive area, we can say you have the situation selection, situation modification. In fact, you have and attentional deployment for attention, cognitive changes, and response modulation. Now I just want to say different emotion. You can see the basic emotion. Finally, it's really automatic processing, and not really really automatic versus elaborated emotion. And in fact, why I take that? Just because I think the surprise, it's more automatic processing, and the fear, for example, it's more elaborated processing. And Plutchik, now everybody can say I know Plutchik's wheels. Thanks, Nathalie. He suggests 8 primary bipolar emotion. Surprise, sadness, disgust, anger, anticipation, joy, trust, and fear. And really interesting, it's joy vs sadness, anger vs fear, trust vs disgust, surprise vs anticipation. But the anticipation for Plutchik, it's more it's leads to preparation and is opposite to surprise. And what's okay? And you can see in 3D because it's really and model of 3D dimension. And finally, he proposed to see the emotion with the primary emotion, and in additional different components, you can see another emotion, and finally, you can feel every emotion can meet on the point, because every construction of emotion takes a lot of different primary emotion. For example, you have surprise and fear. It's give away. After we can propose to see the Ekman coding facial expression for an emotion. You can see the fear emotion. He finally find the real contraction of muscle, facial emotion, facial muscle, sorry, and we can see the fear, anger, sadness, happiness, disgust, and surprise. So we can see we can see finally in the human behavior the fear and flee. We can see on the next on the left pictures, And finally, we find this behavior in animals. It's same for mice or other animals. And why we can use the animal model just because we can't really regard the structure and take the little the smaller thing, the smaller area in the brain. We can't analyze the tissue, the brain tissue, humans. The animal models of psychiatric disorders, because I will present you the different experiments in psychiatric disorders, need to have the criteria of validity for validate the human study. In fact, we need to have the same. Not same, but the same behavior, the same reaction, etc. We need, after, to choose the species. Why species? Because we need to find finally what animal can use for the study, and finally Scherer say the species must have behavior, emotional expression, the same emotional expression, physiological component, cognitive aptitude associated with the emotion, and connective bias, and the logistic systems in genes, nervous system, monoamines, system neuroendocrinian. Evolution and phylogeny. Sorry, I don't translate my teaser, but it just for say finally how to say the mice, for example, it's good model for studies the disorders, the human disorders, psychiatric disorder. In fact, one study just take every component and regard on if the rodent have the capacity to respond of this of human disease. And you can see finally you have the primate, the monkey primate, and mice, and everything or just some different things is not really clear, but it's really good model for studies the human disease. And I come back on the facial expression because in fact Berridge finds the facial expression on rodent, and you can see finally the expression of disgust on the newborn. It's same finally the rodent and primate, and you have the positive to sweet. It's same. You can find finally the facial expression, not every facial expression, but some few facial expression. After, we can see the gene analysis. In fact, one recently paper of CB letter, it's analyzes the human gene expression and the mice gene expression, and it's really correlate for the structure, different structure of interest. So now I just want to present you the UCMAs, unpredictable chronic mild stress. What's the unpredictable chronic mild stress? It's finally to respond of depression disease, human depression disease. And for that, you have two really important symptoms. It's apathy and anhedonia. And in mice, we can see the depressive-like state, and we have the not really same, but the analog behavior with spontaneous grooming behavior, spontaneous motivation, and appetence for preserved food. It's apathy and anhedonia can find in the mice. Now I just want to present you how to induce the depressive-like state of mice. We need to stress the mice with different stressors, social stress, cage changes. So there's the change without sawdust and over. But it's really important to say when you stress the animal it's finally similar to human depression disease. With the stress is an important factor to develop the depression disease. And I just want to present you, to show you the one really important component of this UCMS. It's first state, because it's finally the motivation to clean, and you can see on the left the control mice, normal mice with condition normal, and depressive-like mice, and it's really visual. You can appreciate. After, I just want to present you two different behavioral tests. The Startle Reflex, representative for me, on the measure of the surprise processes, and the fear conditioning, because the process is very well studied and understood in the neuropsychobiology. And we can imagine that the proximity of the surprise and fear in the primate emotion reveal some similar neuromechanism. The startle reflex, it's used in the human disease, and this is the principal indicator of this test. It's finally the electromyogram on the orbicularis oculi. Just the muscle really fast to respond of startle reflex. Now, I want to show you an experiment. It's just the method. It's participants were shown 9 brief film clips, and in the first 3 clips had no affective valence and were used only to familiarize subject with the procedure. The last 6 were arranged in 2 free clip blocks and shown in the following order: 1 neutral, houses object, and positive it's comedy turn, negative it's toy surgery, and neutral another neutral, it's street scene. Another negative, it's a gangster dragging a terrified victim into the forest. The last positive clip, it's a dance sequence. For the last six clip, we put 3 acoustic stimuli during 50 milliseconds, presented on the 92 decibel in the 92 decibel, and it's a white noise. And I will show you the result because it's really interesting. We don't have any effect between the control group and the patient group, but a different pattern of startle modulation was present in the low and high depression group. In fact, the depression disease have multi-component, and the anhedonia and anxiety make part of it. Now, if we observe the result, the low depressed patients are more anxious and less anhedonic than the higher depressed group. Startle reflex can be indicate the state of depression finally, because we can say if low anhedonia is more expressed for this patients, can be low depressed and high anxiety again. Now I just show you the same procedure for, not really same because now it's not a clip video, but it's startle reflex on rodent. Finally, just we put the rodent in the box. We induce in fact we can measure one contraction to contraction body. Now I just present you different method for this startle reflex with animal. The sensitization, it's finally we put one sound. You have you can see you have a startle reflex, the reflex, and if after we try again to put one sound, we can see finally less activation of startle the reflex. And if we put the foot shock, we can rebuild finally the reflex. Sorry. Now we can use the startle reflex for fear potentiation with conditioning, and we conditioning the mice to see the light signal, and after receive the shock, and after 4 or 24 hours later, we just put on the sound. We have the normal reflex, but after again 4 or 24 hours later, we put the light and the sound, and we can see finally the really more expression of startle reflex. After we can induce habituation. The habituation is same. We put the sound, and after we put another sound, but just a few moments after, and we can see finally the habituation is normal. You have less reflex for the second sound. we can have pre-pulse inhibition, really interesting phenomenon. It's finally we put the sound, and after we put one sound, and just after 30 or 500 milliseconds, we put a real sound, real cues of startle reflex, and we can see finally the inhibition it works. We can use the startle reflex in pleasure attenuation. Sorry. We put the light and the sound. We have the startle response, and 24 hours later, we put just the light, and in fact, it's finally the reward. And another day, we put the twice in this, and we can see the startle reflex, startle response. It's really low. So now I just want to show you the real result with a basic startle reflex with animal models, the UCMS, and you can see different diagram. It's in fact you have the different souche, different species of mice. And in fact, we can see the really interesting. It's this thing, this species, because it's really a good response on the UCMS. You can see the first state. The first state, it's really significant, less bad, just sad, and we can see the startle reflex. It's correlate of depressed-like states. After, I just want to propose and really complicate pictures. In fact, it just I don't want to see you to read you anything. It's just to say the different processing between fear potentiated and pre-pulse inhibition. In fact, we can see in fear potentiated the principal pathway take different structure, but this hippocampus, the structure hippocampus, it really implicates in memory. And in fact, we can say we can have an hypothesis. It's the startle response in fear potentiated calls the memory processing. And pre-pulse inhibition, it's really not same process. It's really really fast and really simple way you can see finally. I'm sorry, the darky box indicates the principal pathway. I just want to present you the future of future techniques and new techniques in animal models. It's optogenetic. It's really interesting because we can study the cognitive process by separation. We can really separate different cognitive process. In fact, the method it's we take the mice gene, the mice promoters. We put this in the virus capsule, and we inject that in the brain of mice. And finally, this virus can induce the activity, the change, the activity of neurons with the photosensibilization. And after, if you put the the fiber optical with the light, we can induce, just activate the circuit of the neuronal circuit. I just want to show you, because it's really famous and visual things. Just want to see you an example. It's one we inject the virus in the brain, but we focus and we take the target, the neurons, motor neurons. Sorry. And if you look the video, we can see the normal behavior of mice, and we light on. You can see finally that she can't do another things to running, and we activated the behavior. And after, if we switch off the light, she stopped this behavior and normally life. I repeat if you don't really see. Now it's activated, and she can't do another thing. And after it's finished, and retake the normal behavior. I just want to show you another experiment. It's on the not startle reflex, but reflex. In fact, in mice, it's a really complicated neuronal circuit, and but in Drosophila, I think it's that. Drosophila, we can finally identify the neural circuit for the reflex. And if you put the gene and the virus, we can, with the light, activate the reflex to flight. It's really, I think, it's really interesting. And she has the reflex and not do anything about it. Why the Optogenetic can do another things. Really famous article on this year. It's to induce finally the false memory. What the method? It's really complicated. I really take the shortcut for you for this presentation. But in fact, we put the mice in the A box, and in another without special medication for enable the spatial memory. And if you put after the mice in B and switch on the light in the brain, we put another in the A and the C, and in fact we active the event memory in B, and we locked the spatial memory during this period. And the result, it's really, sorry. Yeah, the behavior control light for event, it's finally and really and the dental gyrus of hippocampus, the structure of brain. If we regard, if you show, we can see in blue, not really easy to see, but this it's finally the mice with activated virus on the memory, and we can see the activation, the freezing. The freezing. If you put the mice in the A box, in A we don't have freezing, we don't have the fear. After we put in A prime, and in fact we induce the freezing. And she remember as chucked in this box, but not really, in fact, because it's in B. And in fact, we perturbate the memory of mice. And now we just control the freezing. If you light on the structure, if you switch on the light on the structure of of environment, it's another structures, we can see no freezing. It's just for validate the activation of behavior to the fear behavior finally. We take the same same method, so but we put another box. It's C box, and we activate or not the event memory. The event memory, it's the shock. It's really fear behavior. And we can see, in fact, the mice really the mice when okay, sorry. When you want to induce the fear, just light on the light, and the mice have an expression of fear. And in fact, it's possible to drive an active fear response with optogenetics. After, we just want to say the same things. Finally, it's possible to generate an internally represented and behaviorally expressed fear memory via artificial means. In fact, the mice don't remember the B box because we locked the memory, the spatial memory, and we just have introduced in memory just the shock, the event of procedure. And if you light on in B prime, it's same box of B, but without shock. If you light off the mice freeze, because the mice don't have received the light on during the shock, freeze if you don't switch on the light in B prime, but if you active the light, the mice don't know. She's really perturbed and don't know if she has received the shock or not. And on the right, you can see in the novel box. And in fact, if you light off the activation of memory event, you don't have the freezing, you don't have the fear behavior. But if you light on, in fact, we induce the fear response on the mice. So I'm sorry, it's really complicated. I tried to too simply, but it's really difficult. Sorry. Surprise, it's my conclusion finally. If we can identify the connective process of startle reflex and isolate this circuit without the other circuit, we will maybe visualize the real pure surprise-like. Yeah, I think it's really an ambitious project, and maybe in future we can induce the surprise, or not really surprise, this surprise-like behavior just with activating neural circuit. And in fact, it's okay. Thank you for your attention, and I'm open for your questions.